This is the model answer video, Key Assessment 3 Resit paper. I hope you've watched the Help Me video where we were just trying to explain what was going on in some of these questions. And here's the answers. So we've got a reaction here, a substrate and a product, and the conversion is catalyzed by an enzyme. And if that happens, we get high blood pressure. This enzyme, we suggested, removes 2 of these amino acids, and we need to sort of say what would be the effect of that. Well, this is our primary structure, so if we change our primary structure, then we've got these possible links between R groups that enable the chain to fold and bend, and that creates the tertiary structure. So really, this is what we're talking about here. When things are binding to each other, it's all about shape. So removing the amino acids, this will change the tertiary structure, or the 3D shape. And now this new shape will bind or not bind depending on what the so the new shape will bind, because angiotensin 1 doesn't bind and angiotensin 2 does bind. So now this new shape, when it's this shape is now different, so new shape is complementary to the receptor shape. Will bind to receptors. It's now complementary in shape. Or the reverse argument, the original one didn't bind. This drug, enalapril, I can't even say that. It tells us it's an inhibitor, so I'm thinking competitive inhibitor, non-competitive inhibitor, I know about that. And it inhibits this reaction over here, so this is our possible inhibitor there. Using the graph, explain how the inhibitor works. Well, we've got to decide between competitive and non-competitive. The big clue here is that as we increase the concentration of our substrate, which is angiotensin 1, then this line approaches the normal non-inhibitor line. So the effect of the substrate has the concentration of the substrate has an effect, which is our big giveaway. So this is a competitive inhibitor. So this name, whatever it is, enalapril, is a competitive inhibitor. It fits, what's the name of the enzyme? ACE. ACE's active site. And we could go on forever, blocking it, substrate can't bind or something. It's only 2 marks. We need to use a bit from the graph, so we can't afford to waffle on. We need to use a bit of the graph that tells us why that's true. The effect is overcome by adding more substrate. So the effect is overcome by adding more substrate, or a concentration of substrate has an effect on the inhibition or the rate of reaction. How enalapril reduces high blood pressure? Well, if this is our high blood pressure down here at the end, our inhibitor is stopping this reaction, or inhibiting that reaction that produces this product that leads to high blood pressure. So less of our product, angiotensin 2, formed. So less angiotensin 2 formed. And that's what causes high blood pressure. Part C, the rate of the enzyme catalyzed reaction is affected by temperature. Explain how. This is an absolute gift. You've got your classic temperature graph on the effect of the rate of reaction. So we just need 6 marks. We need some marks for explaining why the rate's going to go up to the eventually to the optimum temperature, and then why it goes down again. So below the optimum temperature, why does it go up? We've got more increasing kinetic energy. So an increase in temperature increases kinetic energy. What effect does that have? That means more collisions between our substrate and our active site. So more collisions between substrate and active site. This is just straight out your notes. You could say we've got more successful collisions, got more energy, moving faster, that sort of idea. More enzyme substrate complexes formed. More enzyme substrate complexes formed. We always try and put that into an enzyme question somewhere. And we could there's another mark, but you might not immediately think of it. So more activation energy. But I'm happy with those 4 things there. And then above the optimum temperature, so we're now talking, why does this rate of reaction go down? Well, it's going to say generally what's happening. We're going to say denaturing of the the enzyme. What type of bonds are broken? Hydrogen bonds are broken. So the vibrations increase and eventually will break these bonds. Now, what effect does that have? That changes our active site shape, due to the tertiary structure shape change. So a change in that 3D tertiary structure, which changes the shape of the active site, and so the substrate is unable to bind, or less enzyme substrate complex is formed. So substrate no longer able to bind, or it's now not a complementary shape. Less enzyme substrate complexes are formed. Words to that effect.